Nature 2020-24. Today we have with us Dr. C. Vishwanathan, Joint Director ICR IARI New Delhi. Sir will deliver the lecture on genome editing for food security and enhancing climate resilience. Vishwanathan Chinnaswamy has obtained his BSc degree from TNAU and Master's and Doctorate in Plant Physiology from IRI New Delhi. Sir gained his postdoctoral experience from University of Arizona, USA and from University of California. The Sir have vast research experience on how plants sense abiotic stress signal and mechanism of resistance to abiotic stresses. He has contributed a research of ABA signaling pathway, cold stress signaling pathway, and microRNA biogenesis pathway in plant. He has cloned and characterized about 10 different genes involved in abiotic stress tolerance of crop. Sir has done a lot in the field of abiotic stress management in crops. Talking of his research publication, Sir has guided 3 MSc students and 12 PhD students and served as advisory committee member of more than 250 students till now. He has published more than 250 peer review papers in national and international reputed journals and has more than 22,800 citation, which is one of the highest cited plant scientists in India. Sir, I welcome you for today's lecture.
Thank you very much for your kind introduction. Just I will share my presentation. So in this, we have only students, faculty, and what is the pattern of distribution?
Assistant professor to dean.
Okay, so all faculties. Only teaching faculties are there.
BSc, MSc, PhD student bhi hai, sir.
Okay, all are there in this. It is diverse group. So I will try to give an overview of this, and then just I will go through what the progress we have made at this institute. Thank you, Dr. Ankur, and all the organizers for giving me an opportunity to interact with such a large gathering of participants. So, whether my slides are visible?
Yes, sir. Yes, sir.
Is it moving? Is it slides are moving?
Yes, sir. Slides are moving, sir.
Okay. Thank you so much. A very good evening to all of you. So I am from the Indian Agricultural Research Institute, New Delhi, India. And this institute is seat of Green Revolution, and today it is contributing to the sustainable food security of the nation. The institute was started in 1905, and many of you might be knowing. Maybe for those who are not knowing, this institute is started in 1905 at Samastipur district in Pusa, Bihar. And in 1934, a massive earthquake was there. So the building and infrastructure, they got damaged, and then it was shifted to this place at New Delhi in 1936. And this institute has started with 5 different disciplines of research, and today about 26 different disciplines of research is being taken up here. And this institute also offer master's and PhD degree program. The MSc program started in 1923, and in 1958 the University Grant Commission, once it was set up, this was approved as deemed to be university just immediately after Indian Institute of Science, Bangalore got approval. So from last year, we also offer undergraduate program. So we do both research and teaching at this institute. Today I'll be talking specifically about one of the emerging technologies which can contribute to the food security and climate resilience of the country. So this we know that we have to feed about 9.7 billion people by 2050 without using more land and resources while lowering the greenhouse gas emission. So also we are facing the problem of micronutrient malnutrition. So we have one side challenge from the higher demand, and the other side the climate change is impacting our production and productivity. It is putting a lot of stress on the farmers who are involved in the production side, and also it is going to be a major challenge for the future generation. And the United Nations has also set certain sustainable developmental goals. Some goals are very related to agriculture. One is the zero hunger. So the entire sustainable development goal is depends upon how efficiently we produce food in a secure and sustainable way, and how we can disseminate or deliver it to the consumers at affordable price. So the agriculture plays very important role, and also it plays important role in the climate action, as we have to reduce the greenhouse gas emission, and agriculture is also one of the contributor, although not a major contributor, but it is one of the contributor for greenhouse gas emission. We have to produce sustainably, minimize the greenhouse gas emission. And the poverty alleviation is highly related to agriculture, because about 50% of our workforce or the population is engaged in agriculture. So if we increase the agriculture productivity, then only we can alleviate the poverty, because many poverty is highly there in the rural areas, which can be definitely alleviated by proper agriculture, because that is major source of their livelihood. So if we increase the agriculture income, then it is going to contribute to the poverty alleviation. So in several other sustainable development goals, agriculture is going to play very important role. So as we know that we have achieved a lot in the food sector, that is agriculture has made tremendous contribution to the independent country, and today if you are such a great nation, and the contribution of agriculture is one of the important factor. We could provide food all the people, and we should thank our policymakers and farmers and scientists who made it possible. With only about 2.4% of the land resources and 4% of the water resources, we are supporting 18% of the global population. So this is the 2047 target that Niti Aayog has fixed for us to meet the growing population demand, nation's need. When we celebrate our Amrit Kal, that is about 100 years of independence, we need to produce about 520 million tons of food grain. Currently, we are producing in 2023, it was 324, now it is 333. So we have to produce about 520 million tons. So about 190 million tons of additional food grain need to be produced. And in fruit and vegetable also, we need to increase the production significantly. So if you see other the milk and everything, we have to, the target is very high, about 1.5 to 2.5 times we need to increase the productivity. At the same time, we have to reduce the post-harvest losses, and we have to increase the water use efficiency. We have to increase the nitrogen use efficiency. These two are most important input that are used in the crop husbandry. So we need to increase the efficiency. So this everything is possible only if we use frontier technologies, and we plan our research and development in such a way that the farmer can produce high amount of the agriculture production from the existing 144 million tons of the net zone area that we have. And if we include the even the double crop area, about 194 million hectare of grass cropped area is what we have. So from this, we have to produce almost 1.75 times of food and fruits and vegetable production need to be done. And in addition to this, now the another competitive sector is the biofuel. As we know that our government policy to mix with the petrol and diesel about 20% and 5% of bioethanol respectively. So this needs additional agriculture produce that will be going towards the bioethanol production. The major sources for the bioethanol production is the molasses and followed by rice and maize grains. These are used to produce the first generation ethanol. So which means more food grains are going towards the ethanol demand also. Already recently you might have seen in the newspaper there is a lot of conflict between the feed industry and ethanol industry for the same maize grain that is available in the limited quantity. So we need to increase the production of the agricultural crops in a sustainable way while within this same available land, while reducing the resource use to a limited extent. So if you see the agriculture production, all the biologists know that the phenotype, or in this case, we can say it is the production, it depends upon the genotype and environment and management. How the genotype or genetic composition is interacting with the environment and the management practices is going to be deciding the productivity. That is the basic equation that governs the productivity or phenotype of the crops that we grow or the livestock that we produce. So everything, the productivity depends upon the genotype, environment to management interaction. And the genotype, the existing genotype, we by using breeding program, we continue to improve the genotype. From the time of domestication, we have improved a lot, and we have improved the productivity, use efficiency, nutritional content, everything we have improved, and we need to continue to improve this. In addition to that, the crop management is another important component, the agronomic management of crops, how we manage the inputs for the crop, how we manage the plant protection aspect, and how we manage everything. It depends upon that the productivity is going to come. So we need to focus on both the aspect. Today's topic is related to genetic improvement. As I told, we have to produce more. We have to produce more. One of the options is to increase the genetic potential of the crop, so that with the available land, available input, even with the reduced time, we can produce crops that can produce more productivity. So when we talk of the productivity enhancement, that is, we are talking about the amount of the yield. It may be grain, fruit, or the sugar, whatever is the economic yield. That can be produced per unit area, per unit time, per unit amount of input. So for this purpose, we need to increase the genetic gain. That is rate at which we are improving the yield, and the related trait need to be increased. So currently the rate of genetic gain is about 0.9 to 1.3% in the major crops. That is, if you see the rice, wheat, maize, and soybean, these four crops supply about 66% of the calorific requirement of the global population. So in these four crops, if you see the rate of genetic gain, it is about 0.9 to 1.3%. So for 2050, to achieve that requirement for 2050 or demand for 2050, we need to double the genetic gain. So genetic gain equation, if you see, this is the Breeder's equation. All the agriculture student might have studied in their breeding course this equation. So the Breeder's equation says how we can increase the rate of genetic gain. So we have to follow this equation to improve the genetic gain. So the genetic gain depends upon the selection intensity. That is, we have a large set of population. From that, we have to select an individual which is highly superior, highly productive. And then for this, we need selection accuracy should be very high. That is, we think that this germplasm is very good, then our accuracy should be very high. Otherwise, if we don't have any accuracy, if 50-50 option is there, then there is the selection is meaningless. So we should have accurate selection procedure. and then we should have adequate genetic variation. The genetic variation comes from the spontaneous mutation or from crossing two diverse genetically diverse plants crossing together. We can create genetic variation. These are the, or you can create induce mutations to create genetic variation. Today we are talking about one of the technique which is known as genome editing, which helps in precisely inducing the genetic variation. So this one is genome editing is one important tool towards creation of genetic variation, which is shown in the equation here. And other one is we have to reduce the amount of time that we take for breeding a crop. So the conventional crossbreeding approach, it takes a lot of time in developing a variety. And also when we do the crossbreeding, there is always like specific transfer of genes is not possible. So multiple gene combination happens, and from that we select the one genotype that is like what we have identified as having the desirable phenotype. So if we use genome editing technology, we can easily correct the genetic defect, or we can improve one or two traits specifically, so that the rest of the character is not change. So if you have a high yielding variety and if it is having disease resistance, only we can edit to import disease resistance. So that is possible. So time required is less by using genome editing technology. And other one factor which is reducing the genetic advancement is the inbreeding depression. Inbreeding depression is nothing but when we are trying to cross two different genotype to bring the good character together, because the genes are linked with each other, like within a linkage group. When they are present, when a good gene and a bad gene is present, we are bringing everything together. So this causes an inbreeding depression. And also this is like by breeding, when we introgress genes from one genotype into another genotype, this kind of negative allele can come, that can negative genes can get accumulated in the recurrent parent or the recipient genotype, where the genetic gain is reduced. So in this case of genome editing, we are not going to cross any genetically diverse genotype. Only the existing genotype is edited. The gene sequence is modified in a specific way to produce the improved plant. So this possibility is not there. So in all the way, genome editing can create genetic variation, it can reduce time, and it can minimize the deleterious allele accumulation. Another advantage of genome editing is that if we know one particular pathway is conserved in different crops, so if we have to use the conventional genetic approach, we have to cross two genotype to bring the genes together and to select the combination that is more efficient. But we know that if a gene that is giving a specific trait that is identified in rice, because all the cereals genome is conserved, so we can by using the genome sequence analysis, we can identify the ortholog from the wheat, and we can modify that wheat gene directly by using genome editing. So there is no we cannot transfer the superior allele from the rice to wheat, but we can directly edit the wheat allele by using genome editing. So this is another advantage, that the similar allele can be created across the gene pool where the breeding is not possible. So much advantage is there for the genome editing technology. So genome editing technology is a precision mutagenesis tool. It can create mutation very precisely. So why mutations are important? And everybody knows that mutations are the basis for evolution, and mutation is the basis for creation of genetic diversity. And here I have given two examples, two mutation example I have given. So one mutation where Professor Norman Borlaug standing in the wheat field. This is the normal wheat, which is having the normal allele of reduced height 1 gene, which encodes for a DELA protein, which is involved in the VA signaling. And this dwarf wheat that is produced because of the mutation in the reduced height 1 gene. So this plant is GA insensitive. So this plant, the stem growth is reduced. So this produced a semi-dwarf genotype, which will not lodge because of fertilizer. So we can use fertilizer and irrigation so that we can produce more from the same unit amount of land. So this led to green revolution in wheat. This gene was transferred from Marrington genotype, which had this RST1 mutation. The another one is the mutation, dwarf mutation in rice. This short semi-dwarf mutation in rice. The SD1 is a mutation in the GA biosynthesis gene. And this mutant version was identified a genotype called as DGOgen, and this was transferred to a tall genotype to produce the miracle rice or IR8 genotype. So again, here a single gene mutation that produced the semi-dwarf rice varieties, and this wheat and rice mutation, these two mutations, they created the green revolution in these crops, and this helped in providing food security to the nation. Otherwise, in 1960s, it was expected that as per the Malthusian theory, the population growth is too high and food grain production cannot be increased to that extent. So population reduction will be there because of the hungry-induced mass deaths. So that's why that hypothesis or that theory was proved wrong by these two mutations. So you know what is the power of mutation. So only you have to create such kind of mutation by using the new technique. So these mutations were the spontaneous mutation that was discovered by scientists long back. So that's why scientists try to always, they were interested in creating, seeing that if we can create mutation, that is the frequency of spontaneous mutation is very low. It may be about 10 power minus 6 to 10 power minus 9. So scientists were trying to create the induced mutations by using various approach. This is one example of the earlier discovery of induced mutation. So Hermann Muller and John Stadler, they were working on the induced mutation. Dr. Muller was working with the flight Drosophila, and Stadler was working with the maize crop. Although he started his work earlier, Stadler started his work earlier, and Muller started little late, but he used Drosophila as model system. The Drosophila, as you know, it has very short lifespan. Only in 12 days it can complete the lifespan, whereas maize, it will take long duration, 5 months, 4 months, and again often in those days maize can be grown only in one season in a year. So his research progress was very slow, and his progress was faster. So he could demonstrate that X-rays can induce mutation, and mutations are heritable, and he got a Nobel Prize for the same. So later, there are several mutagenic agents have been identified. You can see sometime here I have listed many of them. It may be ionizing radiation, or alkylating agent, or DNA intercalating agent. So many the chemical and physical mutagenic agents are listed here. These mutagenic agents, when the genomic DNA is exposed to these mutagenic agents, these mutagenic agents create DNA damage in different places in the genome. So when the damaged DNA is repaired by the native repair mechanism, some mistakes happen that leads to the mutation. So again, the mutation caused by these mutagenic agents, they are random, and it is unpredictable with and rare with reference to a specific target gene. But it will maybe creating a large number of mutations it will create, but maybe the what we are interested, those kind of mutation in that specific gene may not be available. So that's why still biologists were looking for the way in which we can exactly create mutation in the genetic code of the organism. So there comes the contribution of these two professors, Professor Jennifer Doudna and Emmanuel Charpentier, who discovered the RNA-guided genome editing. So this technology came in 2012. Before that, there were earlier technologies were there for the targeted mutagenesis. So these three technologies, one is the engineered homing endonuclease or the meganuclease, which is a native nuclease enzyme. It binds to certain sequence in the native organism and it makes cleavage. So it is a DNA binding nuclease. So all the DNA binding proteins, they recognize a specific sequence depending upon the amino acid composition in the DNA binding domain. So in the meganuclease, if you modify certain nucleotide, the nucleotide, and if you produce a meganuclease enzyme, which can bind to regions of the genomic DNA, which is newer regions. That is, if you want to target the meganuclease to a specific gene, then we have to engineer this protein so that it can be targeted to any other location where it can go and recognize that sequence and make a cleavage. So purpose of this one is to make DNA break. As we told that all the different mutagenic agents, they create DNA damage. So when it is repaired, the mutations occur. So in the same way, these nucleases are used to create a DNA damage. So now we can target these nucleases to a specific location by modifying the DNA binding sequence. So this one again, it was very difficult because the nucleus domain and the DNA binding domain, they are in one protein, and when you try to modify the amino acid sequence, the secondary and tertiary structure changes, so it may alter the activity and DNA binding properties. So it was very difficult work, and only sophisticated labs which are capable of protein engineering, they could do it. And another one is the use of zinc finger and nuclease combination. Zinc finger is again DNA binding protein which has specific DNA binding domain. So if we assemble 2-3 zinc fingers or more zinc fingers, we can target the zinc finger complex to a specific location in the genome. To which your nucleus, endonuclease is added. In this case, mostly POC1 nuclease is used for this purpose. So this combination of DNA binding protein plus nucleus was used for certain times. And then the transcription activator like effectors were identified. These are the bacterial transcription factors. When bacteria such as Xanthomonas oryzae infect rice, they inject this tail protein into the cell, and these tail proteins go and bind to the host plant gene promoter, and they activate the expression of susceptibility gene. So these are the bacterial transcription factor, but it activates the transcription in the plant. So they have certain DNA binding domain which can be easily engineered to target the tail effector to any gene of our interest. Again, to this tail effector, if we combine the nucleus, it can the protein complex will go to a specific location, and then it can make cleavage in the genome. So all three techniques, they are sequence-specific nucleases, sequence-specific protein, plus nuclease that are designed to target specific location. So with this method, since protein engineering was involved, many laboratories were unable to do it. At the same time, it is very difficult to engineer this protein to target the loci or place where we are interested. So in this context, this RNA-guided genome editing came in, where instead of the DNA binding protein that decides the place where the nucleus is going to cut, here in the RNA-guided genome editing, we use an RNA molecule that takes the nucleus to the specific location, and it helps in cleavage in that particular location. So this technology was published in 2012 in the Science magazine. By 2020, it has revolutionized the field of medicine and agriculture, and Jennifer Doudna and Emmanuel Charpentier, they received the Nobel Prize for Chemistry in 2020. So what is this technology? As I told, the mutation basis for mutation is that DNA damage. So DNA damage has to be created only in one specific location, so that there only the mutation is created, rest of the genome is clean. So for this purpose, you need two reagents. One is the molecular scissor or the endonuclease that cuts the DNA in the specific targeted site on the genome. The other one is the RNA molecule that takes the molecular scissor to the specific location on the genome. So these two reagents must be delivered into the cell. So this two reagents can be delivered as DNA, or it can be delivered as RNA plus protein. DNA means that is the DNA which codes for the single guide RNA, that is the 20 base nucleotide RNA molecule, which is complementary to the target site, and a scaffold RNA which helps in binding up the Cas9 enzyme. So that is it is called a single guide RNA. And the Cas9 is the endonuclease, this protein. So this is again in the DNA sequence, we put this coding sequence so that the single guide RNA and Cas9 nuclease, they are delivered as DNA into the host, and then once it enters, it integrates into the genome, and then it is transcribed to produce hgRNA and Cas9 protein, and these two form a complex and then go and search a target location and where it create DNA damage. Alternatively, if we can do a protoplast mediated transformation, where we can use the transcribed single guide RNA and the Cas9 nuclease protein. So here we can use only RNA and protein. So here no DNA is involved. So this one is useful when we want to create a transgene-free plant in the vegetatively propagated crop species. So to make this, some details are there. This one is required only for the students, those will be really working in this area, where the single guide RNA is cloned in front of RNA polymerase 3 driven promoter, and which has a terminator which is RNA polymerase 3 specific. So this produces a single guide RNA molecule, and then we express the Cas9 protein either by using 30-phase promoter or by using ubiquitin promoters. So for dicots we use 30-phase promoter, for monocots we use ubiquitin promoter. So the Cas9 enzyme, originally it was used from Streptococcus pyogenes, which is a bacterial enzyme. So bacteria, they don't have a specific demarcation of nucleus. So in case of plants, it has to be transported into the nucleus. So for this purpose, we have to add certain nuclear localization signal. That is, these are some of the amino acid sequence that should be there in the protein. And only this protein will be going into the nucleus. So in plants, all the proteins, they are produced in the cytosol. After that, depending upon the tag it has, some of them they are put into the nucleus, some of them will go to chloroplast or mitochondria or to the plasma membrane. It all depends upon the signal peptide or signal sequence these proteins have. So in this case, we have to put nuclear localization signal so that this protein will enter into the nucleus. So the transcribed guide RNA and the protein, it enters into the nucleus. Once it for this purpose, the delivery of the reagents, it can be done either through Agrobacterium or particle bombardment mediated transformation, or if we are using protoplast, just we can use the protoplast transfection. So once these reagents goes into it, the Cas9 and the single guide RNA complex, it search for the homology sequence. And one important thing is that the Cas9 needs a protospacer adjacent motif, that is 3 or 4 nucleotide sequence, conserved sequence, where only it can bind. So this one normally in case of Cas9, it is NGG, and if it is for CPF1, it is AT sequence. And for different enzymes, we have different protospacer adjacent motif. following the proteospecial adjustment motif, the 20 nucleotide target sequence is there. This we design. That is, if you want to transfer our the Cas9 enzyme to any location in the genome, we have to accordingly design this 20 nucleotide single guide RNA, and this 20 nucleotide guide RNA is cloned in this vector to produce the sgRNA complex. So once it binds, it create a double-stranded break. That is, it has a cut site on both the DNA strand, the Cas9 cuts, and it will create a double-stranded break in the DNA strand. So once the double-stranded break is happened, then the work of the Cas9 and single guide RNA is over. So now the cell takes over the repair mechanism. One of the repair mechanism is non-homologous end join. That is, these two cut ends are try to join these two cut ends, and this gives rise to insertion or deletion. Maybe one or two base pairs are deleted, or one or two base pairs are inserted. The deletion may be little longer also. These kind of mutations are called as site-directed nuclease 1 mutation. So this will come to the latter part of the presentation, where we will be talking about SDN1 and SDN2, which are exempted from the GM regulatory rules, so that it can be cultivated normally. So in case of other approach, once the cut is made, we can tell the cell how to repair the cut site. So for that, we need to give a repair template. That is, the cell to copy, we need to provide some template. So the cell can copy whatever is the genetic code written there, it can copy into this place. So the repair template is homologous to this the cut site. That is, it is exactly same as that of the cut site DNA sequence, except that it may have few nucleotide difference, so that the new template, it can be by homology dependent repair mechanism, it can be incorporated into the cut site, and we have the new mutation introduced. So here we can precisely decide if you want to change one nucleotide, we can introduce in that one. So this is called as site-directed nuclease 2, where after the cut is made, we provide a repair template to get the repair done. And the third kind of mutation is the SDN3 mutation, where we provide a foreign DNA flanked by the sequence which is similar to that of the cut site. So now by HTR, this foreign DNA can be incorporated into the cut location. So this one is useful when we don't have genetic variation. That is, if no gene is there, we cannot do any editing. So for example, if want to put BT gene in cotton, so we need to bring the BT gene from the Bacillus thuringiensis, which is not available in the cotton. So for this purpose, we need to take the gene from the Bacillus thuringiensis. We have to integrate that gene in the cotton genome. So for that, the advantage of SDN3 over the transgenic is that we can insert that foreign gene in a specific location where the expression is good and it is not going to disturb any native gene. So these are called as SDN3, and SDN3 mutations are now considered as GM crop at present in India, but in USA and other countries it is not considered as GM. So in addition to this traditional approach, there are two advanced approaches are there. So this one, I think some of you might be working on genome editing, and those who are working will be knowing about this one, where the other approach is called base editing. That is, if we know that the deaminases are there, which can remove methyl group from the nucleotide. So if we use this deaminase fused with the Cas9 enzyme, once it goes and bind to the specific location, and the single-stranded cut is made, instead of double-stranded cut, here we mutate one nucleoside in the Cas enzyme, so it can create only one strand cut. And the deaminase that we use deaminate the target nucleotide. It may be either the cytosine or it may be adenosine. the cytosine deamination, which results in the production of uracil, which is then converted subsequently become T. So it creates GC2 AT mutation ultimately when it is finally the mutation is arrived. So the C is converted into T. In case of adenosine deaminase, again the adenosine is converted to inosine. Inosine can mimic C, so it becomes C2. The A to G mutation arises. That is, CG site will arise in the place where the adenosine was deaminated. So we can create an AG substitution here. So this type of substitution is possible by using base editing. The other advanced version is prime editing, where again once the cut is made, we can give a copy template which is RNA instead of DNA. So an editing template is added to the single guide RNA. So this one desired number of nucleotides are changed. So this one will bind to the cut site and the new strand is synthesized by a reverse transcriptase. So in this, the RNA is also long. That is in addition to the sgRNA. At the end of the sgRNA, the editing template is given. So this is not known as pegged RNA. That is prime editing guide RNA. And then with the Cas9 enzyme, yeah, reverse transcriptase is attached. Here, deaminase is attached. So in this case, yeah, reverse transcriptase is attached. So this can also create desired change specifically in the genome. So these are different technologies that are commonly used. There are some other advanced versions are being emerging. So I think these versions are the most common, and this will be there for a very long time. So when we talk of the gene editing, often we think that just we can delete some gene or make the gene non-functional. So it is not so. We can create any kind of mutation. So those who have studied biology, we know that the gene consists of a promoter and a 5' translated region and 3' untranslated region, and it consists of exon and introns. So once the gene is transcribed, which depends upon the promoter, that is, we have maybe rice genome has about 40,000 genes. All the genes will not express in the all the time, depending upon the tissue, depending upon the environment condition. A set of genes will be expressing at that specific time and in the tissue. So that is determined by the promoter. How it is deciding the transcription? Because the promoter has a cis element. These elements are the sequences that are to which the transcription factor binds, and then the transcription factors once bind, they recruit the RNA polymerase 2, and then they produce transcript. So this cis element sequence can be modified so that a gene can be altered for its expression. So for example, if fungal infection induces a susceptibility gene, you can delete the cis element so that the fungal cannot infect. So there will be no induction of the gene. But in all other conditions, the gene expression will be normal. So that kind of modification is possible, and the upstream ORF editing is possible. Upstream ORFs are small protein coding sequences that are there in the gene, and this attract the ribosomes. That is, once mRNA is produced, ribosome binds on this URF, and they translate a small protein, and then they follow. So because of this, normal ORF is not, protein is not produced much. So if we remove this URF, the normal protein production will be there. So we can increase the production, and we can also create exon mutation to specifically modify the protein function or sensitivity to herbicide or sensitivity to various stresses that can be modified, so we can produce efficient enzyme. And also we can edit the microRNA binding site so that we can create microRNA resistance or susceptible version. So gene editing is a versatile tool which can create either knockout, altered expression, or altered activity, altered sensitivity type of gene expression variation. So genome edited mutants are non-GM crop. because in case of transgenic, we use a foreign gene, and foreign gene is integrated into the genome of the target crop. And finally, we select for the plant which is having the foreign gene in its homozygous condition. And whatever variety that we release will have the foreign gene inserted into it. And the commercial variety will have this foreign gene. And if we do a molecular analysis, we can detect that this plant is having a foreign gene. So these are transgenic. In case of genome-edited crop, so the foreign gene gets integrated. That is, as I told, the Cas9 and guide RNA, they are put into DNA sequence. The coding is put into DNA sequence, and once the transgene is integrated into the genome, the single guide RNA and Cas9 is transcribed, and then the transcribed single guide RNA and translated Cas9 protein goes and bind to its target location, and then it creates the mutation. So this mutation and the tDNA, it can be segregated in subsequent generation. So the final product is consists only of the mutation of the tDNA, which we used for genetic transformation. So the mutation in the commercial variety, no foreign gene is there, and we cannot like by using any technique, we cannot say because of what reason the whether it is created by a natural mutation or it is because of the genomic technique that we have used. So this mutation is similar to that of the mutation that we get because of the conventional approach. It is same as that of the conventional mutant. That's why the mutant that we are generated or considered as non-GM, and it is similar to that of the natural mutation that occurs. So that's why the genome-edited plants are considered as non-GM in diverse part of the globe, which are shown in the green color. In Europe also now you can see some green spots. Soon they will also accept it. As this technology has a lot of potential for agriculture sustainability. So already the genome-edited product are there in the market. One is the soybean, genome-edited soybean, which is marketed in USA from 2019, and the gamma-aminobutyric acid-rich tomato, which is marketed in Japan, and the fish that are edited to produce higher biomass and produce more protein within short period of time. These are available in the market from 2021 in Japan. So in our laboratory, we started work in 2017. We work on diverse activity, and today I'll be just presenting about one case study what we have done. So this is one of the mutations that was discovered in 2009. A paper was published by a Chinese group of scientists. That is, if they mutate one specific gene, which is called as drought and salt tolerance, a zinc finger domain containing transcription factor. The loss of function gene mutation, it gives tolerance to osmotic stress and salinity stress, and also to the drought stress. So from that time we got interested in this gene. As that time since beginning of my career, I was interested in working on developing stress tolerant plant. So we searched the mutant population that was available with us, which is Nagina22 mutant population, and in this population we searched and we could not find any mutation in DST gene. So even if you know the gene, you will not be able to get the desired mutation so easily. So this work was started with in 2018. Dr. Santosh, who is working with me as a postdoc, he has started using this technology to edit a specific gene. And the initial guidance and the construct that are required, we received from Professor Jiang Zhu. He was my mentor in my postdoctoral program. So by using this approach, as I explained in the earlier slide, we delivered the guide RNA which are specific to the DST gene, and we delivered the Cas9 into the callus of rice cultivar M2U1010, and then we regenerated the plant. And if you see the kind of mutation that we obtained after sequencing the putative transgenic plant, we can see that there is a mutation that is, this is the guide RNA sequence that I have underlined, and this is the PAM site that I have shown. As I told, the Cas9 will bind to this place, and guide RNA will bind to this region that is underlined. And normally the Cas9 exactly cuts 3 bases upstream to the PAM site. So at that cut site, the repair happen, and there is a mutation in the cut site. So here we can see a single base deletion or 2 base deletion mutation, or when the DNA is cut between the 2 cut site, the whatever region in between it is totally removed and perfectly joined. So these kind of very perfect mutation can be created by using this technology. And because of this mutation, and you can see that there is a change in the protein sequence, because from the site where if single or 2 bases are deleted, you know that there will be a frame shift mutation. This frame shift mutation can result in the non-functional protein. The idea was to create a non-functional protein. So we could create different kind of non-functional protein, and then we confirmed by Sanger sequencing and in different generation, and also we analyzed whether this produce any off targets. These analysis also we have done, and based on the predicted off target, we sequenced all the regions that is possible off target region. So whenever we design a guide RNA, it can bind to the genome in multiple places, but it may have a few mismatches. Those regions are called as the off target regions. So these mismatches here in the predicted off target, they are shown in red color fonts. So this indicates that these are in the guide RNA, it is different, and in these genes they are different. So this kind of few nucleotide mismatch can also the guide RNA can bind to the DNA loosely, but the Cas9 will not cut if these mismatches are there within the 12 base pair upstream to the PAM sequence. So when we design guide RNA, we can see that there are some mismatches in the regions off target in the guide RNA when it is binding to the off target. If some mismatches are there in the seed region, then off target will not be there. So when we select guide RNA, we have to select such guide RNAs which have some mismatches to the off target region. So we for both guide RNA we predicted, and then we sequenced those regions from the plant. That is, we have used genome editing to create specific mutation. So if you want to ascertain that the phenotype that we are seeing, it is because of the mutation, not targeted, not because of the off-target mutation. So for this purpose, we sequenced the predicted regions, and we found that the sequences in the predicted off-target sites are same as that of the wild type. So there is it indicates that there is no like off target mutation is there in the mutant that we identified. And then we examined the phenotype, and we found that this mutant they use less water, and then we examined stomatal conductance and the actual number of stomata present. These are the wild type, and the arrow indicates the stomata, whereas in the mutant we can see less number of stomata, and also it uses less water as compared to that of the wild type. Then we also analyzed for the reasons for this, and we identified some genes which are involved in the stomatal development. They are less expressed in the particular mutant. And we can see that the mutant plant, they are more surviving under the wild type plant when we use sodium chloride or when we use PEG. And then in the earlier publication, they have shown that it can tolerate seedling stage, but we were interested in checking at reproductive stage stress tolerance. So in this case, we examined reproductive stage stress tolerance, and we found that as compared to the wild type plant, the edited plants, they produce more number of grains under stress condition. We also examined their stress tolerance at under the drought stress. These are severe dehydration stress that laboratory experiment where the plants grown in the soil, right? They are like exposed to no watering or drought stress, and the mutant plant they grow better and survive better as compared to the wild type plant. So these are the plants, the wild type plant and the mutant plant that are grown in the same pot and exposed to minus 90 kilopascal stress. And the survival rate of the edited plants, they are better than that of the wild type plant. Then we also examined whether the growth of these plants under normal condition, and we found that under normal condition, the edited plant they grew better and they produce more number of panicle and it produced more number of grains, and ultimately they produce the total number of fill grain per panicle and high grain yield per plant. That is, edited plant they produce more grain as compared to that of the wild type plant. Then we have also examined them under the NATO's transgenic NATO's stress condition, where the edited plants they produce higher grain yield under drought stress condition as compared to that of the control plant. However, in the field under normal condition, also these edited plant produce more yield. So these plants they produce more yield under normal condition and produce higher yield under drought stress condition also. So now, once we have the mutation, now we have to get exemption from the rules 1989. These are the rules enacted, developed under the Environment Act of 1986. So we have to get exemption from these rules if you want to take this genome edited plant into the field for field testing and further release into the farmers' cultivation. So in our country, the genetic engineered plants they are approved by a different committee. Some important committees are Institutional Biosafety Committee, Review Committee on Genetic Manipulation, and Genetic Engineering Appraisal Committee. So if you want to, if you are engineering a plant by transgenic approach, you have to take the approval of the Genetic Engineering Appraisal Committee, then only it can be released into the environment. Whereas for the genome edited plant, as I told, they are not GM crops. So yes, different set of rules were formulated. So in this case, the new guidelines were brought in by the government of India, where if the SDN1 and SDN type of mutation, if we show that they are free from the introduced exogenous DNA, that is transgene that we use to deliver the Cas9 and guide RNA, then this can be exempted from the rules 7 to 11 of the rules 1989. That is, once you show that the mutant plant is free from foreign DNA, then it can be considered as normal plant. So for this, the standard operating procedure was developed, and then based on this, we can do all the experiment to show that it is not containing foreign DNA, and then we can get exemption. So towards this, we have done the experiment for our mutant, and we have shown that the transgenic plant from which the final genome edited plant was obtained, we have to use these plants, and then we have to use the wild type plant or the original variety in which the transformation was held. And in genetic transformation experiment, we use a selection marker. So in this case, we used hygromycin as selection marker. So we have to show that the transgenic plant is tolerant to hygromycin, whereas the mutant plant is not containing the foreign gene or hygromycin gene, so it should be as sensitive as that of the mutant. So here you can see that the last column and the first column, which is wild type, and the mutant that we develop, they are equally sensitive to the hygromycin, whereas the transgenic plant was tolerant. So this indicates that the mutant plant is not having the gene that is used for hygromycin for selection. And then to show that no foreign DNA is present in the mutant plant, we have to design the PCR primers to cover the entire vector sequence that we used for genetic transformation. So we designed about 49 different pairs of the primers to cover the entire vector sequence. So the condition is that the amplicon size of each primer pair should be 500 base or less, and the subsequent the amplicon should have a 50 base pair overlap at least. So by meeting this condition, we have carried out this experiment to show that the wild type plant and mutant plant, they do not contain any foreign DNA, whereas the transgenic plant or the wild type plant DNA with spiked with the vector DNA, where the amplification is happening. That is, the PCR conditions are okay with that particular primer where it amplifies, whereas the wild type and the final edited mutant, they do not amplify that specific amplicon. So that indicates that these amplicons, that 500 base pair amplicons, they are not there in the genome edited final plant. We can see in all the cases here no amplicon is there, and in some cases you will also see no amplicon is there in the transgenic plant, because you know that when we use agrobacterium mediated genetic transformation, most of the time only the tDNA regions get integrated. So if the primary is from the tDNA region, you will find an amplicon. If the primary is from the non-tDNA region of the vector, you may not find the amplicon there. By using this approach, we obtained exemption from our IBSC and RCGM. So this was one of the earliest mutant that obtained RCGM permission and is in the active field trial. We expect that next year, depending upon the result, we may release it for cultivation. So besides that, we are working on other things for improving the yield. We have developed a mutant which can produce higher yield. And this is another one mutant which is developed by one of our collaborating institute, Indian Institute of Rice Research. From there, Dr. Satyendra has developed one mutation in Samba Masuri background, which again produces a very high level of 25% higher yield as compared to that of the original Samba Masuri. And in National Rice Research Institute, they have developed a mutant of the Swarna, which can produce high yield as compared to that on the original line. So this line again, next year it may go for field testing. This line is already in field testing. So after the success of this, we are working in various other crop in a network project mode. And based on the success of our work, and the government of India has supported ICR with 500 crores for editing various crops. So this project is running in 54 different institutes, about 24 crop science institute and 24 crops for insects, and about 10 horticultural institute on 50 crops and 40 genes. And then with other animal science institute, poultry, animal sciences, livestock and fisheries, all these institute, about 53 different institutes are working under this project. So this project started in the March 2024, and we hope many products will come in this one. And we have also discussed with Professor Jennifer Doudna, the Nobel laureate for the genome editing, to sign an MOU with ICR so that we can train our scientist student in their lab in the advanced areas of training. And we have also initiated another work on genome editing for bacterial blight resistant. Recently, this project, just now it is approved. So we are working on multiple aspects. So to conclude that, we need to use this genome editing technology, and it is very powerful technology to produce climate resilient crops. But ultimately, the research question is very, very important. That is, the planning is very important. The gene you select is very important. The crop you select is very important, and the kind of allele you are creating is more important. So that's why the research question is the one that gets you Nobel Prize. So whenever we plan any genome editing experiment, we need to plan properly so we can develop a product to deliver, or it can be also used as a functional genomic tool to decipher the function of the genes in plant. So finally, I thank all my mentors starting from Dr. T. Mahapatra, former Director General and Secretary there, ICR, and current Director General Secretary there, and Dr. D. H. Sharma, Deputy Director General Crop Science, and D.K. Yadava, ADG SEED, and the Technical Advisory Committee, which regularly monitor our program and guide, and the Empowered Committee of NASF, and all my students and collaborators. So that is in brief. Just I want to say about the genome editing program that we do in our institute and ICR as a whole. So thank you for Dr. Ankur for providing this opportunity to interact with the participants. So now if you have some specific questions, like I'll be happy to answer.
चैट बॉक्स को ऑन कर दीजिए एडमिन महोदय.
Okay.
जी सर.
जो भी पार्टिसिपेंट्स की क्वेरी स्पेसिफिक टॉपिक को लेके है, वैसे सर का बहुत ही बेहतरीन सेल्फ एक्सप्लेनेटरी रहा. मुझे नहीं लगता कि कोई भी क्वेरी की गुंजाइश बची होगी, लेकिन यदि आप चाहते हैं तो आप पूछ सकते हैं.
Yeah, if anyone want to ask any question, like you can tell me. So.
सर का ऑलवेज प्रेजेंटेशन ऑसम ही रहता है. आप प्लीज क्वेरी पूछिए अपनी.
Thank you so much. Thank you so much.
सर तारीफें आ रही हैं सर. ऑसम प्रेजेंटेशन, नाइस प्रेजेंटेशन.
Thank you. It is good that you are organizing this seminar for large number of participants. At least even at this time, about 250, more than 250 participants they have joined. I think it is good initiative. You are doing very good work. It's good that many people can get exposure to different emerging topic and important areas of agriculture.
एक्चुअली सर 30 दिन एकोमोडेट करना एक अपने आप में बड़ी चैलेंजिंग बात हो जाती है, तो उसको ऑनलाइन कर देते हैं.
Yeah, definitely it is very yes. This somebody is asking PPT. I will send my PPT to you, so you can share the PPT to the participants. Okay?
जी, thank you so much.
So there is one question from Dr. Neeti Sharma. That was, can we use genome editing for viral resistance?
Virus resistance.
Yeah.
Which one? Genome editing for virus resistance. Yes, genome editing for virus resistance can be used. So normally, people have shown that in the translation elongation factor, if you make specific mutation, we should not make a loss of function mutation, but if you make a specific mutation, we can bring in resistant to viruses, which has been shown in rice and other crops. And also in tomato, there is a gene called Plicora where we can create a specific mutation that give resistant to virus. So it is possible to create resistant to virus and the fungal disease and bacterial disease, which has been already shown in different crop. In case of insects, also for the BPH and the stem borer and several other insect, it has been demonstrated. So for all these things, you should know what gene has to be mutated or edited, what modification is required. So already if some studies are there, people have studied that this gene is a susceptibility gene or this gene is a resistant gene. If you make some modification, the resistance can be imparted in the alike variety. So it is possible to create virus resistant. Some of our institutes are also working on viruses resistant. Even in IRI, we have a program in which they have started the viral editing in the new project for resistance to virus in the new project that was started. So
another question is from
yes, please.
Yeah, another question is from Arshi. Why CRISPR is not widely usable in practical way?
I think this is one of the technique that can be widely used. So it is like PCR maybe after few years, because currently a lot of expertise is not there. This require at present the expertise in genetic transformation. So because in between for several years, not many research laboratories, they were working on the genetic transformation, because we were not like commercializing the genetically engineered crop. So anyone interest in this is limited. Only those who are interested in studying the gene function, they were doing the genetic transformation. So genetic transformation is one of the limitations. So if any lab they can do genetic transformation, they can do gene editing. Because we also started in 2017, and we started at ab initio that thanks to Dr. T. Mahapatra, he believed that we can do it. So he gave a project, you try. So without any training or anything, we started, and many laboratories, they started in the same way. And almost everybody who has expertise in genetic transformation and recombinant DNA technology, they could do it. So it is in practical way, it is like widely useful. One limitation currently is the IP, that is it is under the patent rights of the University of California, Berkeley, and Broad Institute, MIT, Harvard. So this one now government is in the process of negotiating with the IP holder, so that once you develop product, the product can be taken to the farmer. So maybe after like now, when the molecular breeding was started, only few laboratories or one or two laboratories they were using. Now, if you see several breeding department, they use molecular markers for marker-resisted breeding. So this technology is one technology which is going to be widely used in maybe in a few years. Maybe it will not take much time. Maybe in 4 or 5 years, you can see most commonly used. And we are also, ICR is in big way, it is using this technology to develop new product. And the DBT Institute, they are working on it. And the NIPGR has developed the mustard, which is having less glucosinolate, and ICGB has developed product in rice which can have more yield and more iron and zinc. So many institutes are working. Nabi, Mohali, they have developed banana which is rich in provitamin, and many institutes like TNA has developed aromatic idli rice. So there are several examples are there. They are coming up. So some of mutants are in advanced stage, and some may come next year field trial. So we expect many product will come within very short period of time.
I think this is well explained about the question. Another question is from Angana ma'am, and that is kindly inform the status of BT brinjal at present in India.
So BT brinjal is under the moratorium. That is government banned cultivation of BT brinjal. It took all the approvals were obtained. That is the all the biosafety tests, everything was done. Finally, the GESC approved it, but the Ministry of Environment and Forest and Climate Change, it imposed a moratorium. That is still they clear it, or still the government orders we should not cultivate the BT brinjal. So BT brinjal is cultivated in Bangladesh. In India, it is not cultivated. In India, only cultivated transgenic crop is the BT cotton. So different gene stacks of the cotton is only cultivated crop, and it produces a lot of fiber, and it also produces a lot of edible oil. So that edible oil we are using, and most of the edible oil that we import from other country, whether it is soybean or it is canola oil, they are all from the GM crop. So nothing wrong in using this, but it is a perception of the people, and also necessity. Like maybe government think that BT can be the brinjal can be like managed with conventional approach, because it is not a major like food crop, so they might have thought that it can be managed with the conventional approach. Still, a real demand arises. So currently, the Brassica, GM Brassica is under debate, and although the GESC cleared, and again it is with the Supreme Court, the final verdict is yet to come on this one, whether we can cultivate the GM mustard. So it all depends upon the policy and the public perception. So we need to create awareness among the public, like the GM is not this anything bad that is going to create. All the drugs that we get, recombinant drugs in India, they are all by using the same recombinant DNA technology. We are getting cheaper drugs without a need for importing. So if we have to be Atmanirbhar, we have to use multiple technology. Because GM food are the one which are highly tested. That is, you test the crop in diverse places, you test for all its character and also toxicity, allergenicity to the all the environment and to the human. So it is well tested product. So there is no harm in consumption, but it may take some time the change of public perception.
Thank you so much, sir, for clearing all the doubts of our students, faculties, as well as scientists who are listening to your words very patiently. I think your lecture was self-explanatory. I, Dr. Jyoti Sharma, on the behalf of AgriMeet Foundation, as well as on the behalf of World Agriculture Organization, thank you for the today's session. We are very thankful that you have spare your valuable time for our session. We hope to have your lecture in future also and try to have your support, blessing and motivation further in other program also. Thank you so much, sir, for today's program.
Thank you so much. Thank you all. Thank you, Dr. Jyoti, thank you, Dr. Ankur, and all the participants. Thank you very much. Thank you.
Good night, sir. Bye.
Yeah, good night.
Thank you, sir.
Thank you.